live with the North Carolina Museum of Natural Sciences in beautiful downtown Raleigh. It's Science Tonight. Now here's your host, Chris Smith.
Good evening, everybody. Hey, folks. Welcome to Science Tonight. I am your host, Chris Smith, brought to you by the North Carolina Museum of Natural Sciences. Thanks for tuning in to tonight's program. Glad that you're all with me. Hey, over there in the YouTube chat box, drop like a waving hands emoji or a say hi. Let me know that you're here checking out the program. As we go throughout tonight's show, you can also leave your own insights or questions for our guest speakers in the chat box. Later in the program, I'll be grabbing those cues so that we can get some answers from tonight's guest experts. We do have a pretty exciting show for you because most of the time we just have like one guest expert on the program. Tonight, I've got three for you. Let me introduce them now. Dr. Kevin Kocot is... Oh, no, I have the wrong one. Dr. Kevin Kocot is an assistant professor in the Department of Biological Sciences at Alabama, the University of Alabama. Kevin, welcome to the program.
Thank you very much. Happy to be here.
He's also the curator of invertebrate zoology at the Alabama Museum of Natural History. So look at that. We've got some cool museum connections going on in tonight's program as well. I'm excited about that. Also joining tonight's discussion is Caitlin Reddick. Caitlin is a PhD student at Auburn studying marine invertebrates. Caitlin, welcome to Science Tonight.
Thank you for having me.
Glad you could be here. And a name that fans of the North Carolina Museum of Natural Sciences will definitely know, Megan McCuller. Megan is the collections manager for non-molluscan invertebrates at the museum. Hey, Megan.
Hey, happy to be here.
Yeah, good to see you again. Now, everybody, I have the three of them on the program today because just a few, really days, not even weeks, really, the three of them got off a boat having been in the frozen hinterlands of Antarctica for 3 months or so, doing research on, if you hadn't guessed it already, invertebrates, the spineless frozen critters living close to the South Pole. Is that a fair description of Antarctic marine invertebrates? They're like, yeah, I think so. It works out for this case. So I'm excited to learn more about the trip to Antarctica, why you wanted to study Antarctic invertebrates in the first place. But as is standard for Science Tonight, we're going to have a little bit of fun first. So Kevin, Caitlin, and Megan, are you familiar with the sea shanty craze that took the internet by storm?
Yes.
Yeah, definitely. So what I've done is I've prepared a sea shanty Mad Lib. We're gonna go one by one. You're gonna give me nouns, verbs, or other figures of speech, and then we're gonna see what your sea shanty is.
Oh gosh.
I love it.
Does that sound all right? Since you just got off of the boat not too long ago, sailing halfway around the world, I thought sea shanties would be a perfect way to start off the program. So the way this is going to do it, I'm going to go just around the horn here, tell you what I need from you when I call on you. So I'm going to start with you, Dr. Kocot. I need a noun.
Mollusk.
Mollusk, of course. Caitlin, I need another noun.
Dog.
Dog. Very good, very good. And Megan, a third noun.
Isopod.
Isopod. All right. Kevin, back to you. A verb ending in ing.
Trawling.
Trawling. Very thematic here. All right. Caitlin, another verb.
Dig.
Dig. Oh, this is going to be great. And Megan, a noun.
Oh man, I was expecting a verb.
Ooh, I'm sorry.
Oh my gosh. I'm totally blanking. Boat.
Boat. Perfect. All right, just 4 more. Kevin, a noun, please.
Now, Antarctica.
Ooh. Okay. Caitlin, I will need another noun.
Grilled cheese.
Grilled cheese. Megan, you get to give me a verb this time.
Digging or dig.
Stick with that one. Okay. And the last one will go to you, Kevin. A direction, like up, down, left, or right.
Down.
Down. All right. Now, I won't make you sing the sea shanty, but I will read it off to you so that you know what you got. Now, this is based, I pulled the traditional lyrics for one called Wellerman. This is apparently like the sea shanty that was out on TikTok and everything. So here we go. Wellerman by Kevin Kocot, Caitlin Reddick, and Megan McCuller. Here we go. I'm not going to sing it though. I'm just going to read it. Soon may the Wellerman come to bring us mollusks and dogs and isopods. One day when the trawling is done, we'll dig our boat and go. That sounds really nice. I like that. And then next verse, there once was an Antarctica that put to sea. The name of the Antarctica was the grilled cheese. The winds dug hard, her bow tipped down, blow, me bully boys, blow. Round of applause. Excellent sea shanty. Somebody go back in this video and put it to music and then send it to me. And we'll play it next week on Science Tonight. Check that out. Now, okay, I'm looking this over. Excellent job. And I have to think that the appearance of Antarctica, boats, and lots of invertebrates, except for the dog, are top of mind for you for the program. So perhaps we should get into there. Although is grilled cheese related to being on a research boat in Antarctica?
It was the best thing to eat in the dining hall when you got off a shift, and they always had them. So I was thinking.
So grilled cheese was it. So let's get into it. Let's talk a little bit about this research trip then. Kevin, Dr. Kocot, you were the principal investigator for the trip. So you were one of the people in charge of this expedition to Antarctica. So give me and the viewers a sense of what this trip was about and what its purpose was.
Sure. So I was one of 5 of the different principal investigators on the cruise, so I can't take all the credit. The chief scientist was Ken Halanych at Auburn, and he and I worked together a lot for a lot of the planning. But trying to put all the different pieces together for the cruise had to begin really far in advance in terms of planning what gear was going to be sent down, who was going to be able to go. And so there were a lot of logistics involved, but for someone who's never done this before, it's my first rodeo planning a cruise. It was a bit intimidating, but the National Science Foundation and the Antarctic Support Contract were super helpful to kind of get everything organized and did so much of the behind the scenes work. So our jobs were made as easy as possible for planning a 3-month-long cruise. And so a lot of packing, shipping things down way in advance, making sure things got there safely, and making sure we had everything. So once we got down there, we couldn't go back and get it.
Yeah, once you're at sea, there's no just putting in at Ecuador to pick up some snacks, right? And so what was the overall purpose of the trip? Why head to Antarctica in the first place? What were you hoping to do?
Yeah, so there were 5 different projects. And my project, for example, was focused on the biodiversity and systematics of one particular group of marine mollusks. They're called Apocrines mollusks. And I realize that's not a normal like dinner table name, but the reason we care about focusing on taxonomy and understanding the diversity of this group or groups like this in general is because there's a real need for training for the next generation of taxonomists who can go and identify organisms like this. And so This project is focused on understanding the evolution of this one particular group, but also bringing down students who are going to be hopefully the next generation of taxonomists who are learning how to be an expert in invertebrate biology and whatnot. Some of the other projects, for example, Dr. Halanich's project, the chief scientist, was focused on the biodiversity and biogeography of Antarctic marine invertebrates, looking at population genomics. So I think Caitlin will probably talk a bit about that later. Other projects were focused on another group of invertebrates called humations or comma shrimp. Another project was focused on the microbes living in the sediments in Antarctica and sort of their biogeochemistry. So a lot of different projects all kind of going on, and then we also had some side projects as well, but the focus was really on kind of the two main groups of invertebrates we were working on and the biogeography.
Okay, very interesting. So, Caitlin, you were working on population genetics. Is that what I heard right?
Yes.
What made studying population genetics in marine invertebrates in Antarctica different or special? Why go to Antarctica? That's what I'm trying to get to.
I was like, well, it's in Antarctica. I mean, I guess one thing that was really interesting for me about it is a huge part of population genomics and general population dynamic research is having appropriate sample sizes and getting kind of exactly the numbers that you need to do the type of statistics that we want. But when you're in Antarctica, you can't really be that selective, right? So it was definitely a bit of a Wild West adventure. But it was very exciting. I liked it. It was fun to kind of go in, and a lot of the research that we're going to be doing now is kind of dependent on everything that we were able to find. And I got to be exposed to so many new different types of animals and groups of animals that I could kind of, play with and learn about. And so it was, I mean, just a really great experience. Who wouldn't want to go to Antarctica?
Yeah, where was my invitation? I know where it was lost in the mail.
Yeah, we got lost.
I'm also not an expert on marine invertebrates like the three of you are, so I couldn't have expected one in the first place. So what was did you have a specific role on the ship? I mean, if you're an expert in population genetics in these marine invertebrates, was it to, point and say, that's one of those? What was the job?
Well, my job specifically as kind of like a graduate student was a pair of hands more than anything else, I would say, which it was great. So we were separated into two different teams, and each team kind of had a team leader. So Kevin was kind of the head of his shift, and then Dr. Halanych, who was in Canon, who's my PhD advisor, was the head of my shift. And so basically what would happen is they kind of would tell us what to do, and everyone else on the team just was all hands on deck, literally, at any given time. But for my lab specifically, I did get to take on a role of being like, okay, we need this many of this animal that we pulled up, and we need this many of this animal. And so just kind of making sure that we got everything that we needed, and and making sure that when things were coming back, we knew where they were, and so on and so forth, and just kind of organizationally helping out.
Gotta have help. And expert help. You said just hands to do work, and I'm thinking but they're experts in invertebrate zoology to begin with, so that's
I mean, when we all got on the boat, I would say our expert level was very different than when we got off. We all felt a little bit we had quizzes that we would go through and test and make sure we were all up to speed on identifying things, and so there was a lot of kind of we all started pretty junior in what we knew about all the invertebrates that we were going to see, but it's a fast learning curve.
And so Megan, the invitation came to you, I guess, being a part of the museum. How did you get connected with this opportunity to sail to Antarctica?
That's a good question. I guess my name is just sort of known now in circles, as somebody who does bryozoan taxonomy, so a certain kind of animal that's colonial and very common in marine environments that not a lot of people study. And I was actually contacted by Kevin, Dr. Kocot, to be an alternate. And that's just because there's this physical qualifications process that you have to go through, everybody has to go through. They were being a little more strict due to the pandemic. And I ended up going. I ended up working with Dr. Halanych, though, and that worked out well for me as much as I would have liked to be on Dr. Kocot's group. Dr. Halanich's group was the way they were sampling, like the equipment pulled up animals that were more likely to have bryozoans on them, or just pull up bryozoans themselves because they're larger.
So you were helping identify just the bryozoans that were coming up, or another set of hands like Caitlin?
Both.
Both.
Yeah, the major part was just like helping pick and sort when the samples would come up, and the other was just processing and general identification of all the invertebrates. But I did do the bryozoans as well, and that was one of the things that would happen sort of at the end, since we were trying to get like certain groups of organisms processed before others, just because of the priorities of the grant.
And so, Kevin, tell me a little bit about some of the, I guess, the species. What critters were you pulling up? Because I guess I'm from the sea shanty, we got trawling as the verb. So I imagine you were out in the ocean around Antarctica, and then you just haul up nets full of big, little invertebrates and then sort through them. So what were you hoping to find? What were some of the interesting ones that you did find?
Yeah, so we were primarily trawling with what's called a Blake trawl. So it's sort of a net on a metal frame you can drag behind the ship to trawl up invertebrates. And there were pet groups that everyone had, and there were particular tasks that were written into Halanych and Andy Mahan's grant, for example. But really everything that came up was considered, and kind of our philosophy is fix first, ask questions later, because once these samples come up, they're probably not going to survive if we throw them back overboard. And so we basically photographed everything. We fixed at least tissue samples of, if not the whole individual of everything. Because these samples are hard to get, even if they're things we don't have plans for, another researcher might. And so they're going to go to one of these museums and be available for anybody to access if they need it. Particular groups that we were focused on in my lab are mollusks. We work on snail, slugs, clam, squid, octopus, and these weird worm-like apocrines mollusks that I mentioned. But we also like to work on any kind of tiny invertebrates. And there's a lot of groups of animals that are, all of them are tiny. And so when we're looking under the microscope, that's kind of an opportunity to find a lot of things that aren't really well known or even new species. So we would spend a lot of time sieving the mud that comes up in some of the trawls, where the other team was picking up all the big charismatic megafauna, the large conspicuous animals. And we're hunched over a microscope getting seasick as we're watching our samples do this under the scope. But we found a lot of really exciting things doing that. Small, but exciting.
Like what? What are some of the, are we talking just weird shaped worms? Are they worms that you wouldn't see in more temperate waters, like off the coast of North Carolina? I'm imagining so, but not really certain what you would see down at the bottom of the earth.
So yes and no. There were like a lot of familiar groups where we see their relatives in temperate regions. Some things that are kind of rare taxa that are at least more diverse in the Antarctic and not common in other parts of the world. And then certainly, new species in all of these groups that, I shouldn't say new species, but Antarctic specific species that, we don't have around here, for example. So familiar things and a lot of really strange things as well, but lots of worms, lots of small crustaceans, so many different kinds of worms, and a lot of things that took us a while to kind of figure out what they were. We saw a lot of strange things. There's a lot of weird miniaturized taxa that are living kind of like in these interstitial environments between the grains of sand and whatnot.
So Caitlin, why Antarctica? What do we hope to learn by spending time?
So for our group specifically, we were interested in the biogeography. And I know I've talked to you about this a little bit. It's people don't know what the land under Antarctica looks like. And so there's a lot of questions about interesting population dynamics that we can do because of that, because these big under ice passages are unknown. And Antarctica is just like a really interesting environment in general, because there's a lot of interesting things that happen with physiology down there. But I think what a lot of people don't know is Antarctica is a huge area. So if you're like me and you're interested in population genomics, and a huge part of that is environmental variability, being able to collect animals in all of these different areas at once is a really valuable way to kind of do research and do science. And on top of that, it is a space that we're going to be seeing a lot of change of in the next, decades. And so being able to go down there now and take stock of what things look like is really valuable. And hopefully we'll be able to go back again and see how things are changing. But it's just, the environment is so drastically different from anything else that you'll see here. So it's kind of the perfect place.
I'm gonna have to back up. You said Antarctica might not be a solid landmass.
That's Kevin smiling. We don't really know. So there's a hypothesis that it's actually a bunch of archipelagos or islands, and that there's underwater passageways that animals can travel under and mix between populations. And so when we're doing these population genomics, the idea is if we can detect signals that there's been breeding between groups that are separated by ice, then we can make conclusions about maybe they're able to travel underneath that ice to be able to make those connections. That is what that hypothesis is testing.
Okay, so your work specifically, you could trawl and look at the invertebrates in one place, and then the boat goes somewhere else. Mm-hmm. And if you see genetic similarities between the two, you could say that they are related, but how did they bridge the gap if they're hundreds of thousands of miles apart? Right. Is that, am I in the right zone here?
Yeah. That's exactly
and so
right.
That would,
the idea is if they're separated by ice, they're not walking across. So there must be some sort of underwater passageway that they're able to get through. And that's what we're hoping to find out over the next however long it takes to process all the samples.
Okay, now also when you explain this, Kevin was smirking. So I'm curious his opinion on this archipelago hypothesis.
Maybe. So this might be something that happened historically. In the past, the ice was not as thick, and so there were connections across different parts of Antarctica. But it's crazy to me that we just don't know that much about the landmass and where land ends and ice begins.
Very interesting. And that's a cool way to try to get at that question too, is to look at these critters that are just hanging out in the water all over the place.
Yeah, it's pretty fun. I can't take credit for it. I entered into a lab that does the cool work. So it's very interesting.
So another thing that I'm curious about, and Megan, I'll bring you in for this one, is actually being on the ship. Because I'm thinking, I saw Megan's tweets about being on the ship, and there's just like gorgeous sunrises and shelves of ice and harrowing experiences of ice breaking that were all over Megan's Twitter account. As she complained about the limited bandwidth for tweeting. She didn't complain, I made that up. But what was life on the boat like? What was life as a scientist on a research vessel?
Good question. I am going to go ahead and share my screen since I did have some pictures.
Oh, excellent. I'm excited to see.
First off, this is a group photo of all the science crew that was on the boat. We actually got to get out on land at one point, which was pretty cool. And we took this photo.
Was it it looks very cold.
It wasn't as cold as you might expect Antarctica to be, but you have to remember that we were in the southern hemisphere at the time, which means that it was like late spring. So it was about like 30 degrees Fahrenheit. And that, depended based on the wind chill.
Not too bad.
Yeah, it really wasn't too bad at all. Like I expected it to be much colder. But So when we got out on land, we, had to go back to the boat. And this is just showing the Nathaniel B. Palmer, the ship we lived on for 3 months.
Wow.
And you see it from this angle and it doesn't look all that big, which was pretty wild. But then you're living on it day to day, and it seems huge. This is a photo. It looks pretty
big there too.
Yeah, when we were on land and it was, out in the water, it looked very small, which was kind of cool. This shows ice breaking, and it was cool because like we would have this trail behind us, and the boat would like ram up against the ice and then sink down on it to kind of break the ice, and then they would have to like go backwards and then ram again. So ice breaking was a very like slow process when we were actually breaking the thick ice instead of just like going through ice that was thinner. And it was very loud. So going through all this was just like, you could just hear it scraping against the side of the ship.
Was that scary at all?
In the beginning it was, just because it was so loud, at least for me. I don't know about everybody else. And then you just sort of got used to it. It was kind of scary when all of a sudden you would feel the ice when you didn't know we were about to hit it. And it would just be like kind of jarring.
You're eating a grilled cheese and then you hit some ice.
That was the other thing, is that it was loudest in the mess hall. So we're all eating and we're breaking ice, and it's just like, just so loud. You could barely hear people talk.
Oh, wow.
Because the mess hall is at the bow and it's on like one of the lower decks. You're right there where the ice is actually breaking.
Jeez.
During actual sampling, when we'd get to a site after transit, we would do multi-beaming. So that's just like sort of sonar to see what the ocean bottom looks like. And once that was done, we would send down this thing, which is a camera in a frame, and there is a weight underneath it. So like whenever the weight would hit the bottom, it would take a photo. And this is called the yo-yo cam. So because it goes up, it hits the bottom and takes a picture and then goes up again, and so on and so forth. And it would do this for a kilometer. And then once that would come back up, we would do the sampling. So that would be a trawl or like an epibenthic sled. And Kevin talked a little bit about those. The trawl picked up larger things, and the epibenthic sled was more for smaller macroscopic or infaunal things.
Mm-hmm.
And
so like sediment.
Yeah, exactly.
Stuff that would be living in the mud. Okay.
Yeah. So this one was getting larger things, and it would go in this kind of bin. So this, I recall this one specifically being kind of muddy, and so it would just like slop out. And then we would get our specimens. This is after going through whatever comes out of the trawl or the epibenthic sled. The big things separated into buckets based on what group of animals they were in. We would separate them into these smaller containers, or just keep them in the buckets if they were large enough. This one on the left is an isopod, and I remember that one was very spiny. You can kind of tell it was spiny, and it hurt my hands.
Oh, really?
Yeah, you can see the spines on its legs.
Mm-hmm.
And then this one in the middle is a gigantic sea star. If I remember correctly, it's like one of the largest sea stars in Antarctica. And then on the very right is a large scale worm.
Oh, I see. It's a worm with scales.
Yep, exactly.
Is that
so we would
these things
be
are
processing
bigger.
these organisms. Oh yeah.
They're bigger than I expected.
Yeah, these are not.
For some reason, I was thinking like small little wormy things, but these critters are They're big.
There were definitely those things also. It's just, of course, I was taking photos of these bigger things.
Right.
And so that was, oh, I do have a picture of Kevin's stuff. This is an Apocrines. If Kevin wants to talk a little bit more about those.
So these are the weird worms.
Behold. Yeah, so they're kind of like a slug, but instead of having a shell like a snail or nothing like a slug, they're covered in little spines. You can kind of see it almost looks fuzzy. That's because it's covered with little calcareous spines made out of the same stuff a shell is made out of.
So instead of a shell, they have shell material, but made in little pieces like this.
Yeah. So
what
some
do
are
these?
spiny. Sorry.
No, go ahead. I think you're about to answer my question.
So some are spiny. So there's like kind of the porcupine flavored ones like this one, and then others are scaly, so kind of like a pangolin. But so it's like a coat of armor or like protective spines to help protect them from getting eaten by predators.
What do Apocrines do, or what? What is their role? You would say in the environment, their niche.
Everything they do slowly. But most Apocrines feed on cnidarians, so things like corals or hydroids or even like dead jellyfish that have fallen to the bottom. And so they seem to be really picky eaters and only eat like one kind of coral or one kind of hydroid. And then there's some that burrow in the mud, and we think just kind of eat whatever, maybe an animal or an organism called foraminiferans, a protist. We know some eat nematodes. A lot of what we know about their diet is because we've actually like caught them in the act of eating, but we try to sequence their DNA. And sometimes we get like what they had for lunch instead.
Now, for one like we're seeing here in this photo, what's the scale here?
So this is a giant one. It's like 3 centimeters long. So this is with a macro lens on a pretty zoomed in macro lens and a camera. A lot of them we would take photos with a microscope rather than a camera. But some of the biggest ones are actually quite large. Like the biggest one in the world is like over a foot long.
Oh, wow. Okay. So a very diverse group of critters then.
Yeah, they can be like so small they live between grains of sand or fairly large.
Very nice.
Did
we have more? Oh, okay, I was going to say, I was like,
the last photo.
What's the next cool critter to look at? Okay.
Nope, that was the last one.
And so what was it like being on, you're talking about being on the ship and the size of the ship, but what was the work like? So Megan, you and Caitlin both talked about sort of like being on these work crews for processing the specimens that were trawled in. Caitlin, I don't know if you can you break that down for us?
Yeah, it was hard work. It was definitely like, I would put it under like manual labor. But so we would get like these trawls, as you saw in that photo, and there might have seen like the big kind of wooden box and that everything would get dumped into. And then basically everyone who was on shift at the time would come out onto the back deck, and we would just have to be sorting through animals pretty much as fast as we could. And depending on how big the trawl it was, this would take everywhere from like 20 minutes to like 3 hours. So it could really, the variability of how long you're going to be out there is it's pretty big. And if it was a muddier one, we would go and start with Kevin's group, start like sifting through. And that was a lot of kind of shoveling and then hosing and shoveling and hosing, and so on and so forth, and getting really excited when something cool came up out of the mud and it like emerges. So then everything is, as Megan said, sorted into these big, basically 5-gallon buckets, and rushed inside. And the idea is to like we had these big books that had pictures with all the identifying names on them, and we would go through and kind of label everything that we could as fast as we could. And then there was someone who worked at the museum in Auburn, who had what we called the book, which was like the most important part for us. And she would basically point at an animal and tell us exactly how to preserve it and exactly like how many and how she wanted and in what type of preservative, like, is it going to be frozen? Are we putting it in ethanol? Is it going in formalin? And so she was kind of running the whole show. And then once you got your marching orders, you take your animal and you run it over to, we have like these big camera stands essentially. And it gets its picture taken. So it's a little like photo day. and then it immediately is preserved. It's like notated in the book and then preserved immediately. And so we would do that for as long as it took to get through the trawl. And I know for Kevin's group, it was a little bit different. Kevin, do you want to speak to how you guys processed things?
Yeah, it was similar, but everything's smaller. So we kind of had a choke point at the microscopes where we would bring things in instead of sorting them outside on deck. We'd bring them in and look at like a subset at a time under the microscope to pick out all the individual animals and trying to keep our samples on ice while they're in the lab so they stay happy longer.
What were some of the most interesting things that came up? Like we saw some of the cool pictures, but like Caitlin, you're talking about like going through samples and going through and sorting things out. Was there anything that, you sort of like brushed the mud aside and like, oh my gosh?
This wasn't something, so we actually didn't keep this because it wasn't an invertebrate, but we did see a blobfish, which was very exciting.
Fun.
And I know they're very popular. And for me, it was the sea pigs, which are these like
sea pig.
Yeah, they look like little pigs. I think if I'm correct, now I'm forgetting exactly what group they're from, but they're not slugs. They're
cucumber.
They're cucumbers, yeah. So they're these translucent cucumbers that have little antennae and like basically tentacle-like feet, and they look like pigs. I would encourage everybody to Google them. So I think if we ever pulled one of those up, it was like everyone, at least on our shift, like cheered and had to call everybody over to come and look at them. But yeah, so it would, we would just get excited about everything. And then Megan and I would have to cut ourselves off on bryozoan collecting because we would go through and try and get every little tiny shard. And there was one collection where we ended up sorting for like 2 hours because we had just like gone way overboard with what we were pulling out. But it's always fun to kind of just see all the little animals, and we're all really just people who like to play with animals at heart, I think.
Especially sea pigs.
Yeah.
The cutest of the invertebrates?
I think so. I thought they were adorable. Megan?
They're pretty cute.
Well, this is fascinating stuff. In just a moment, I'm going to turn it over to our audience to get their questions and their comments. So see, there's some folks already dropping their thoughts in there, but this is your notice, everybody. Start popping your questions into the chat box because I'm going to start looking there for the questions for the rest of tonight's show. This, when we were chatting about this earlier, Megan, maybe it was you who described this whole thing as a nerd pleasure cruise. Like it was 12-hour work shifts, you told me, but you spend 12 hours just nerding out about invertebrates?
Pretty much.
Doing all the bryozoan collecting, will any of the specimens, since you're the bryozoan expert here, are those specimens going to come back to North Carolina to be a part of our collection, or will they go to Auburn, Alabama? How does that work?
All the samples are taking sort of a long trip, and Kevin might be able to explain more of the process here, but they're all they were on the boat. We had to pack them up pretty regularly just so we didn't have to do it all at once at the end. And they're going to get shipped at some point in the next 2 or 3 months-ish. Yeah, Kevin's like, I don't know.
Something like that, yeah.
We don't really know, but at some point, once they're shipped, they're going to go to Alabama and Auburn, and then at some point, I'll get the bryozoans, some of the bryozoans anyway. So yes, we will be getting some of them.
Exciting. So for each of you, and I'll start with you, Kevin, what would you say was like a standout moment for the research cruise?
I think the first sample we get is always the most memorable. It's exciting to see all these things coming up. Everyone's crowding around. It was really fun for me. The first time I went, I was a student. The last time I went, I was a student, but now I was there with my own students, and it was really fun to see them as excited as I was. That was a very heartwarming moment for me. That was really fun. Then the other moment I remember, which is just funny, is you kind of get laser focused sometimes about being hunched over a table, picking invertebrates and whatnot. And there was one time where I like looked up to kind of like pop my back and realized that I was like next to this beautiful and amazing mountain right next to the boat that I hadn't even noticed. So sometimes it was important to like stop and smell the roses.
Oh, wow. I mean, yes, the background of some of the pictures that Megan shared, just gorgeous scenery. Absolutely gorgeous.
This was the prettiest research cruise I've ever been on for sure. We saw some beautiful sights.
Caitlin, what about you? Any standout, exciting, interesting, memorable moments?
It's similar to Kevin's, but there was one morning where I I was off shift usually, so it never really got totally dark, but we did have these incredible sunrises and sunsets. And I was having trouble sleeping because I was, also in that laser focus, like wanting to know what was going on. And I kind of peeked my head out, and there was like this incredible sunrise, and I ran out, and like everything was icy. So of course I like slipped and mostly fell on my butt, but like, it was just very exciting to kind of be like, okay, yeah, there's all that cool stuff going on, but also look at this. This is incredible. And, be able to kind of take a moment and just like take a deep breath and be like, wow, we're in Antarctica. This is really cool.
Love it. Megan, what about you?
About the same. I really enjoyed going out and just like feeling the boat movement.
Mm-hmm.
I never really did it when there was like higher waves. That's partly because it's dangerous, but it was really neat seeing the sunrises or sunsets, because again, it's like the sun just like dips beneath the horizon. It never really gets truly dark. And yeah, just seeing all the cool animals, and it was just something new every time, it seemed like. I also really liked looking at the yo-yo cam images before the like trawls would come up, just because you could see sort of like what you might get. Sometimes you would get stuff you'd see, and other times you would get stuff that like you didn't see.
Okay.
So that was always cool.
Very interesting. So all that plus grilled cheeses just made for the best experience.
The cooking in general.
The cooking in general?
Yeah.
Oh, okay.
It was just great.
The food was really good.
So, well, now I've got to know about the food. I wouldn't have thought research cruise, the food would have been like top of the line necessarily.
I mean, I had no idea what to expect because I never done anything like this before, but the chefs were from New Orleans, and so it was like a lot of Southern food. There was like barbecue and, mac and cheese, and it was great.
Okay. Maybe the question should have been any standout meals aboard the ship?
I was really passionate about Taco Tuesday. That was like when we were on like in these weird shifts, it was really easy to lose track of time and seeing, and like when you walk in and you're like, oh, it's Tuesday because there's taco. Like it was a nice checkpoint, and also like they just always nailed it. So it was great.
Kevin, did you have a standout meal?
We always had steaks on Saturday, so I looked forward to that. And then Sunday was kind of like lunch on Saturday was steaks, and lunch on Sunday was kind of like Sunday lunch at grandma's sort of thing, like fried chicken or cornbread and all these things. But it was always really good. So that was fun.
Yeah, I know what Sunday lunch at grandma's is like. I know exactly what you're talking about. And it is ever so close to dinner time right now for all of it. Megan, did I skip you? Best meal?
I'm gonna have to agree with them, but they also made cakes for birthdays. So,
oh hey,
and my birthday happened while we were on the cruise, so I got a cake. It was pretty awesome.
You got an Antarctic research cruise birthday cake.
Yeah, it was amazing.
That's pretty special. That's pretty special. Well, this has been great. This is so exciting. I can't wait to learn more, keep up with the results that comes from all of this, what you're learning. But In the meantime, I'm going to grab some questions from the chat box. Ready for that? So let's see. The first one I've got here, John is asking, did you eat any of your catch?
We didn't. I think they frown on that.
There's sea pigs, but there's not sea pork.
We joked about it, but it was no.
Heather is asking, did you catch any vertebrates? Caitlin, you mentioned a blobfish.
Yeah, so I mean, they would come up. It was never the intention, and because of like permitting and grants, we always kind of threw them back over. But there was frequently we would get fish coming up, and we would always, there's some really cool parasites that fish get, so we would always like check their mouths and gills for parasites, and then take anything we wanted off them for the inverts, and then kind of throw them back over and hope that they survived. But I don't think we got anything except for just fish.
So some fish, there you go. Let's see. Earl is asking, this is a good question. Did you hit any bad weather either while in Antarctica or traveling? I don't know who wants to take that one.
Nothing too bad. I remember being warned once or twice that we were going to hit some weather, but I don't recall it being anything that was all that bad.
Well, that's good. Calm seas. That makes it nice. Okay, Anna's got a good question. Gets out a lot of museum work, in fact. Were the organisms killed before being preserved? Can you talk a little bit about the process of collecting?
That's a good question. Yeah, so I always feel a bit guilty when we talk about this because we are killing all these animals. We try to do really minimally invasive sampling, not trawl any longer more than we need to. A lot of the animals are relaxed with magnesium chloride, which kind of like knocks them out and relaxes their muscles before they're preserved. We do that as much as possible. It's nicer to the animals, but also then they don't like contract when they're being preserved. So generally that's what's done, but it depends on the group.
Sure, yeah. And the numbers that you collect are small. Like Caitlin talked about being very precise with how many of what species you want to collect and doing all of the surveying beforehand before you just drag the net everywhere, right. Being precise about it. It's science. Let's see here. Megan wants to know more about the blobfish. At what depth did you find the blobfish?
I don't really remember. It was one of our deeper ones for sure, but I want to say it was like in the 600-meter range, but I could be off about that. It was definitely above 400 meters. It was not a super shallow one.
Glenn is asking, go ahead.
I didn't know about the blobfish. It's the first time hearing it.
Yeah, it was really cute. It was a cute kind of littler one. It was only about this big, but we pulled it up and everyone was like, "It's a blobfish!" And then we all took pictures with it and then sent it on its way.
Oh, nice. That's one of those things that I feel like maybe they're still not real. They only exist in that like one picture on the internet. Nobody has seen one besides that. So it's nice to hear that researchers can, just accidentally pull one up.
Yeah.
Just a big frowny fish. Okay. Glenn has a good question. Do you have an estimate for how many new species were found?
This might be one.
I guess
for
not yet.
Kevin.
Several. I can think of two like off the top of my head where we looked at it and we're like, oh, well, this is nothing that has a name, but there's a lot of things that we weren't able to identify in the field. And I think once we get them back and start looking closely, they might be new to science, but hard to say.
Yeah, we just didn't have a ton of time to really put the most amount of effort into trying to figure stuff out like that. So that'll be in the future once we get the samples in.
Yeah, describing new species is very precise kind of a thing, right? I'm using that word again, precise, but it's like DNA and measurements and comparing it to all of the other species to make extra sure. And doing it on a boat like this probably a little tricky. Okay, let's see. Did anyone get seasick? Kevin's like, yes. Caitlin's like, yes.
You asked about
Megan, you
weather.
said you. Oh, go ahead, Kevin.
Kind of rough at times, and maybe I'm I am a wimp. I get seasick really easily, but there were a few times where I wasn't feeling great for sure.
Was it something that got better over time? Because you're on the boat for 3 months.
We had pretty calm weather for a while, and then it got rough as we got close to Chile, and I was used to calm weather, even though I'd been on the boat, and so then I kind of got sick. And then we were in port for a while in Chile getting loaded up with gear, and then we went south again, and you went from being really calm to like rough weather again. So kind of depends on your acclimation state, I guess.
Yeah, okay. Megan, you made it through, 2 thumbs up.
Yeah, I mean, I sort of took seasick meds just in case. I've never been seasick before, and I didn't want this to be my first time, but I lucked out, I guess.
Nice. All right. Let's see. A viewer named Bronwyn, wink, has questions. Did you find any evidence of plastic pollution?
Yes. Well, I don't know if there was any that was found in like sediments that we pulled up and the cores that they did. But I was out on the bow a few times and saw some plastic pollution, like when we were getting close to Chile. I think I saw like a cup and a bucket and a couple other things that could be spotted by eye.
Oh, wow.
And I'm sure there's probably microplastics, but I think those are going to be, again, things that we won't know until we get the samples.
Right. Yeah. People are everywhere, right? People are everywhere. Bron was also asking, are there research cruise traditions that must be followed?
Yeah, so we do the shrinking cups. We call them shrinky dinks. So it's where you have like a styrofoam cup and you draw on them, and then they're sent down at a deep site, so like 2,000 meters or so, and the pressure of the water makes them smaller. So that's always really cool. And then there was an equator crossing ceremony.
Yeah, keep talking, but Brent,
yeah,
let's
Kevin's
spotlight Kevin.
Yeah.
So that we can take a look at what he's.
Don't judge my art. I can't draw. I get a little deformed sometimes. Sorry, go ahead, Megan. I'm sorry.
Okay, so that was a styrofoam cup that went to the bottom of the ocean and got a. Oh wow, it's so small.
Yeah, I think that's a favorite.
That's cool. So does everybody get shrunken cup souvenirs?
Yes, it was a big thing. Everyone, like, we all make them for ourselves, but then there were some people. Megan, you were one of the people. You must have made like 20.
I made a few. I probably made like
to bring 12. Back to everyone you know.
Yeah. I still need to send them to people. So if you're watching, you might be getting a cup.
Didn't mean to out you for having not sent people the gifts. All right. Let's see. Just a few minutes left here. Let me see what else we got going on. There's a question. How would you compare these icy invertebrates to soil invertebrates?
Wetter.
Yeah, of course. They're in the ocean.
That'd be a good question for Kevin, right? Because like the stuff Kevin was looking for was small stuff, and you are going to find small wormy things in soil. So
Yeah, it's true. A lot of the things that we find in the sediment, have analogs in terrestrial sediments as well. There's lots of burrowing worms and other, critters you would find in soil, and they're at least marine relatives. Like instead of insects, you have crustaceans and things like that. So a lot of parallels, really.
And Chris is asking on Facebook, was there anything you caught that you weren't expecting? Not to bring up the blobfish again.
I have something that comes to mind. We found leeches, and they're on deep sea fish, but there was a leech that was like as long as my arm. It was crazy, this giant leech, and I don't know what it came off of, but
it was terrifying.
I didn't. I don't think you were supposed to find that leech.
I thought they were cool.
Leave that one on the bottom of the ocean. So what happened? So it comes up. Does it become part of the collection? Is it going to be in one of the museums now? Well, that's impressive. So like well, Megan. Okay, I know that our museum has a little bit of a specialty in leeches. Folks like to keep leeches around in their office, for example. So I'm used to seeing these leeches that we have, they're a few inches long. They eat earthworms. And then I'm familiar with like sometimes even smaller blood-sucking leeches, like really tiny ones you get in a pond. But we're talking a big leech. How does a leech get to be like 2 feet long?
I don't know that much about leeches, to be honest.
Okay.
Especially marine leeches. I feel like they were fish leeches, but they weren't on anything when we pulled them off, if I remember correctly. So they were just these huge leeches, just kind of there. Yeah, I honestly don't know that much about them, but I thought they were cool.
I think the name is Piscicodella, which is like fish leech, but that's all I know. They must come on big fish.
Yeah.
That was my next question. I was like, if the leech is this big, how big is the fish? Are they whale leeches?
I don't know if that's a thing. Maybe.
Okay. Well, everybody, tune in to Science Tonight sometime in the near future when I'll be talking with a marine leech expert, because I need to know more about these giant marine leeches. But we've had a great conversation here learning about some of the incredible marine invertebrates that live down in and around Antarctica, and about this very exciting research expedition. If folks wanted to learn more about the activities from this trip or follow updates that will come out of this research trip, how can they do that?
Twitter.
Who should they follow? All three of you? Is that? I didn't know if there was like a trip website or a place where people could click on updates.
Inverts.com, and everybody's Twitter handles is on there, so you can find us that way.
And there's also a hashtag on Twitter, which is #ICInverts, that I think anybody who tweeted on the boat used the hashtag and kind of could use it as a little catalog.
And you said ICInverts.com.
Yep.
All right, excellent. Well, Kevin, Caitlin, Megan, thanks for being on the program tonight and sharing your experiences with us.
Thank you for having us.
Thanks for having us.
This was a great conversation. I hope that everybody watching, wherever you are, enjoyed the show tonight. We'll be back here next Thursday and just about any Thursday night, 7 o'clock Eastern, right here on the museum's YouTube channel. Click that button that says subscribe down below underneath, and you can click the bell to get notified. That way you can tune in to every episode of Science Tonight. We talk to interesting people making discoveries across all fields of science and beyond. Hit up naturalsciences.org as well if you want to see the schedule of upcoming events, this program and others. There's always great stuff coming out of NCMNS. So I hope that you'll be able to tune in to another program real soon. I will let everybody know there's an exciting program coming up on the 18th NASA is landing a new robot on Mars called Perseverance, and the museum is going to have an entire afternoon of live programming to celebrate and watch and tune into the new rover landing on Mars. It's going to be great. We've got our own astrophysicists, local solar system ambassadors who work with NASA. They're all going to be joining us live from the museum. Check out naturalsciences.org. Everything you need to know is right there. And of course, you can get updates from the Museum of Natural Sciences on Facebook, Twitter, and Instagram. We are at Natural Sciences on all 3 platforms. Everybody, take care, stay safe. We'll see you again on Science Tonight next week. Bye, everybody.